Hi everyone, this is Ms. Naranjo. Today I'm going to show you how to fill out the experimental design worksheet for designing your lab on enzyme catalysis. So the first box you see on this worksheet says to identify a problem or pose a question. So since you're going to be doing your lab on enzyme catalysis, your group needs to determine what would be an appropriate question to test enzyme catalysis. In the box that says independent variable, you will be discussing or talking about what the independent variable of your experiment is. In other words, what is the thing that you're going to choose to test for this particular experiment? And so I'll give you a few options in a bit to show you what are some things that you can test. For the dependent variable box, you're going to write down what's going to be the result of you choosing that particular independent variable, so the thing that you chose to test. So keep in mind, when you're doing a graph, the independent variable goes on the x-axis and the dependent variable goes on the y-axis. For the constants, you're going to have to think of a few items that you and your group are going to keep constant throughout the experiment. Remember, the constants are the things that should be kept the same throughout an experiment. They should not change because you don't want them to affect the results of your experiment. If you were to have other items that weren't constant in the experiment, then you wouldn't know if the results of your experiment were due either to you choosing that particular independent variable or due to varying constant in your experiment. So it's important to keep other items constant except for the independent variable that you're choosing to test in this particular experiment. So since you're going to be doing a lab on enzyme catalysis, I'm going to talk a little bit about enzymes. Okay, so enzymes are an example of a macromolecule. They are proteins. So proteins can have many different functions. They can function as enzymes. They can function as immunoglobulins or antibodies, which fight infections. They can function as receptor molecules. They have many different functions within the body, and so enzymes is an example of one way that a protein, one example of their function. So the particular enzyme that you're going to be working with in this experiment is called catalase. Catalase is a protein, an enzyme, that's found in the cells of our body. And so catalase, what it does is it actually takes hydrogen peroxide. Our body actually produces small amounts of hydrogen peroxide to kill bacteria if you do become infected with bacteria. And catalase's job is to break down that hydrogen peroxide because it can become toxic to your cells. So yes, our cells do produce hydrogen peroxide, but it does that to kill bacteria and other organisms that try to invade your body. And catalase's job is to degrade that hydrogen peroxide before it does damage to the cell. Okay, so remember when we talked about how certain molecules fit perfectly with receptors? The same is true of enzymes. Hydrogen peroxide is going to fit in an active site of the catalase enzyme where it will be broken down efficiently. Okay, so if we look at the chemical reaction down below, hydrogen peroxide, H2O2, will be broken down by catalase into water and oxygen gas. Okay, so those are the two main products that you're going to see from this reaction. All right, so some of the materials you're going to be provided with for this experiment, you're going to be provided with a source of catalase enzyme. And so remember, living cells, they produce catalase And that's because it's our defensive mechanism to break down hydrogen peroxide before it builds up in large quantities in our cells. And so your source of catalase is going to be either from potato cells or from yeast cells. Okay, so you and your group are going to have to choose which particular source of catalase you want to use. You can choose between the potato cells or the yeast cells. The other materials you're going to be provided with are hydrogen peroxide, a graduated cylinder for you to properly measure the amounts of liquid that you're going to use, and a plastic pipette for you to transfer the amounts of those liquids. So some of the things you'll be able to choose to test in this particular experiment are pH and temperature. We're going to focus on pH and temperature because those are factors that can affect how enzymes work. So enzymes usually have certain temperatures and pHs in which they're adapted to work best at. And if we stray away from that particular pH or temperature from which that enzyme works best at, then it can actually cause the enzyme to become denatured. So you guys will be able to choose with your group if you would like to test pH or if you would like to test temperature. So if you and your group choose to test pH, some of the items that you're going to be using include vinegar. So remember, the chemical name for vinegar is acetic acid, and hint, it's an acid, so this is going to be your source of low pH. You're going to use water as your neutral pH, and baking soda is going to be your basic pH. So if you choose to test pH, you would be looking to see where catalase is more active, depending on which particular temperatures. Which particular pHs. If you choose to test temperature, what the effect temperature has on catalase, then you're going to be using ice cold water, room temperature water, and boiling water. And so you'll be looking to see at the differences of the activity of enzyme given those different conditions. So keep in mind, one of the things you're going to be looking for or measuring in this particular experiment is the production of oxygen gas. So oxygen gas, you're going to see that as the formation of bubbles in your reaction. Okay, so that's what you'll be measuring. Now back to the experimental design worksheet. Okay, so remember the experimental group, we also call that the treatment group, is the group that's going to receive the treatment. And so the control group is the group that basically does not receive that treatment. So you guys will have to decide which group included in your experiment is not going to receive the treatment. Okay, in the next box below, the experimental design, here you're going to list the steps that you're going to carry out in your experiment. So here on the left-hand side of this box, I want you guys to write out the steps in full detail for how you're going to carry out the experiment. So here you need to be very explicit and specific on how you're going to carry out the experiment. Keep in mind that if you were to write these steps and hand them over to another person, that person should be able to carry out the same exact experiment you did and get the same exact results without having to ask you for any information. They should be able to just read those directions and follow the exact same procedure that you followed. Okay, so you guys will list those steps here, including specific amounts of hydrogen peroxide you're going to use, specific amounts of your source of catalase, the temperature of the water, for instance, or the pH of the solution that you're going to immerse your catalase in, how long you're going to leave your catalase in a particular pH or in a particular temperature before you add the hydrogen peroxide. So make sure to be very specific when you guys list the steps, okay? On the right-hand side here, I realize that those steps might take some more room. So if you run out of room, here on the bottom right-hand corner of this box, I want you guys to draw the setup of that experiment. So how is it going to look when you add all of these items? So if you're going to choose to test temperature, I want to see that you have your graduated cylinder, including those items that you're going to use in that particular experiment, labeled, and how much you're going to add into each variable. So for each temperature, how much hydrogen peroxide you're going to add, how much catalase, what the temperature of the water is going to be, what the pH. Make sure that you draw and label all of that so that you have a sketch of what that looks like. Okay, and I'm going to check your guys' experimental design before we get started with the lab on block day. So make sure that your guys' experimental design is ready to go when you come in that day. On the next page, you're going to collect your data. Okay, so keep in mind that the first column on a table is where your independent variable goes. Okay, so the thing that you're choosing to test, either the pH or the temperature, you're going to list that here. And the second column is going to be your dependent variable. So what happens as a result of you choosing that particular independent variable or treatment. And so you're going to write down the activity of oxygen production or enzyme catalysis activity for that particular column. Okay, and then make sure to have your graphs set up as well. And then we'll talk about this once I get back on block day. All right, so if you have any questions or forgot what I mentioned in one part of the video, just go ahead and rewind the video. Have a good day, guys.